Class 11th Biology, Chapter number 2, Biological Classification. Let's start. Since the dawn of civilization, there have been many attempts to classify living organisms. It was done instinctively, not using criteria that were scientific but born out of a need to use organisms for own use, for food, shelter, and clothing. Aristotle was the earliest to attempt a more scientific basis for classification. He used simple morphological character to classify plants into trees, shrubs, and herbs. He also divided animals into two groups, those which had red blood and those had did not. In Linnaeus' time, a two-kingdom classification system with a plantae and animalia kingdom was developed that included all plant and animal respectively. This system did not distinguish between the eukaryote and prokaryote, unicellular and multicellular organism, and photosynthetic and green algae and non-photosynthetic fungi organism. Classification of organism into plant and animal was easily done and was easy to understand, but a large number of organisms did not fall into either category. Hence, the two kingdom classification used for a long time was found inadequate. Besides gross morphological, a need was also felt for including other characteristics like a cell structure, nature of wall, mode of nutrition, habitat, method of reproduction, evolutionary relationship, etc. Classification system for the living organism has hence undergoes several changes for over the time through plant and animal kingdom have been a constant under all different systems. The understanding of what group organism be included under this kingdom have been changing. The number and nature of other kingdom have also been understood differently by different scientists over the time. Look at the figure carefully and read properly. This is a very important for your examination. R.H. Whitaker in 1969 proposed a five-kingdom classification. The kingdom defined by him were named Monera, Protista, Fungi, Plantae, and Animalia. The main criteria for classification used by him include cell, structure, body organization, mode of nutrition, reproduction, and phylogenetic relationship. Table 2.1 gives a comparative account of different characteristics of the five kingdoms. The three-domain system has also been proposed that divide the kingdom Monera into two domains, leaving the remaining eukaryote kingdom in the third domain and thereby a sixth kingdom classification. You will learn about this system in detail in the higher classes. Let us look at the five kingdom classification to understand the issue and consideration that influence the classification system. Earlier classification system included bacteria, blue-green algae, fungi, mosses, ferns, gymnosperm, and angiosperm under plantae. The character that in un The character that unidentified this whole kingdom was that all the organisms included they had a cell wall in their cell. This placed together group with widely different in the other characteristic. It brought together the prokaryotic bacteria and the blue-green algae cyanobacteria with other group which were eukaryotic. It also grouped together the unicellular organism and the multicellular once. Say for example, Chlamydomonas and Spirogyra were placed together under algae. The classification did not differentiate between the heterotrophic group fungi and autotrophic green plant. Though they also show a characteristic difference in their wall composition. The fungi had chitin in their cell wall, while the green plant had a cellulosic cell wall. When such characteristics were considered, the fungi were placed in a separate kingdom, kingdom fungi. All prokaryotic organisms were grouped together under kingdom Monera, and the unicellular eukaryotic organisms were placed in kingdom Protista. Kingdom Protista has brought together Chlamydomonas cholera, earlier placed in algae with plant and both having cell wall with paramecium and amoeba, which were placed earlier, earlier placed in the animal kingdom, which lack a cell wall. It has put together organisms which in earlier classification were placed in different kingdom. This happened because the criteria for classification change. This kind of change will take place in future too depending on the improvement in our understanding of characteristic and evolutionary relationship. Over time, an attempt has been made to evolve classification system which reflect not only the morphological, physiological and reproductive similarities, but it but is also phylogenetic example is based on evolutionary relationship. In this chapter, we will study the characteristic of kingdom Monera, Protista and fungi of the Viteca system of classification. The kingdom Plantae and Animalia commonly referred to as plant and animal kingdom respectively will be detailed separately in chapter number 3 and 4th. 2.1 Kingdom Monera. Bacteria are the sole member of the kingdom Monera. They are the most abundant microorganism. Bacteria occur almost everywhere. Hundreds of bacteria are present in a handful of soil. They also live in extreme habitats such as hot spring, desert, snow and deep ocean where very few other life form can survive. Many of them live in or on the other organism as parasite. Bacteria are grouped under the four categories based on their shape: the spiral coccus, the rod shape, bacillus, the comma shape, vibrio, and the spiral spirillum. Figure 2.1. Look at the figure carefully. Through the bacteria structure is very simple. They are very complex in behavior compared to many other organisms. Bacteria as a group show the most extensive metabolic diversity. Some of the bacteria are autotrophic. Example, they synthesize their own food from the inorganic substance. They may be photosynthetic autotrophic or chemosynthetic autotrophic. The vast majority of bacteria are heterotrophic. Example, they depend on other organisms or on dead organic matter for food. 2.1.1 Archaebacteria. These bacteria are special since they live in some of the most harsh habitats such as extreme salty area, halophils, hot spring, thermoacidophils, and many and hersey area methanogens. Archaebacteria differ from other bacteria in having a different cell wall structure, and this feature is responsible for their survival in the extreme condition. Methanogens are present in the gut of several ruminant animals such as cow and buffalo, and they are responsible for the production of methane biogas for the dung of this animal. 2.1.2 Eubacteria. These are the thousands of different eubacteria are true bacteria. They are characterized by the presence of a rigid cell wall and a motile flagellum. The cyanobacteria, also referred to as a blue-green algae, have chlorophyll A similar to green plant and are photosynthetic autotrophic. Figure 2.2. The cyanobacteria are unicellular, colonial or filamentous freshwater, marine or terrestrial algae. The colonial are generally surrounded by the gelatinous sheet. They often from the bloom in polluted water bodies. Some of these organisms can fix atmospheric nitrogen in the specialized cell called heterocyst. Example, nostoc and anabina. Chemosynthetic autotrophic bacteria oxidize various inorganic substances such as nitrates, and ammonia and use the release energy for their ATP production. They play a great role in the recycling nutrition like nitrogen, phosphorus, iron, and sulfur. Heterotrophic bacteria are most abundant in the nature. The majority are important decomposer. Many of them have a significant impact on human affairs. They are helpful in making curd for milk, production of antibiotic, fixing nitrogen in legume, root, etc. Some are pathogen causing damage of human being, crop, farm animal and pets. Cholera, typhoid, tetanus, citrus, canker are well-known diseases caused by different bacteria. Bacteria reproduce mainly by fusion, figure 2.3. Sometimes under unfavorable conditions, they produce spore. They also reproduce by a sort of sexual reproduction by adapting a primitive type of DNA transfer from one bacteria to another. The mycoplasma. The mycoplasma are organisms that completely lack a cell wall. They are the smallest living cell known as a can survive without oxygen. Many mycoplasma are pathogenic in animal and plant. 2.2 Kingdom Protista. All single cell eukaryotes are placed under Protista, but the boundaries of this kingdom are not well defined. What may be a photosynthetic protestant to one biologist may be a plant to another. In this book, we include chrysophyte, dinoflagellate, juglinoid, slim mold, and protozoan under protista. Members of protista are primarily aquatic. This kingdom form a link with the other, dealing with plant, animal, and fungi. Being eukaryote, the protestant cell body contain a well-defined nucleus and other membrane-bounded organelles. Some have flagella or cilia. Protists reproduce asexually and sexually by a process involving cell fusion and zygote formation. 2.2.1 Chrysophyte. This group include diatom and golden algae, desmid. They are found in fresh water as well as in marine environment. They are microscopic and float passively in water current plankton. Most of them are photosynthetic. In diatom, the cell wall form two thin overlapping cells which fit together as in a soap box. The walls are embedded with the cilia and thus the walls are industrible. Industrible. These diatoms have felt behind large amount of cell wall deposit in their inhabited. This accumulation over billion of years is referred to as diatomeous earth. Being gritty, this soil is used in polishing, filtration of soil and syrup. Diatom are the chief producer in the ocean. 2.2.2 Dinoflagellate. These organisms are mostly marine and photosynthetic. They appear yellow, green, brown, blue, or red depending on the main pigment present in their cell. The cell wall has stiff cellulose plate on the outer surface. Most of them have two flagella. One lies longitudinally and the other transversely in a furrow between the wall plate. Very often red dinoflagellate example Granulates undergo such rapid multiplication that they make the sea appear red tides. Toxin released by such large number may even kill other marine animals such as fishes. 2.2.3 Euglenoid. Majority of them are freshwater organisms found in the stagnant water instead of a cell wall. They have a protein-rich layer called pellicle, which make their body flexible. They have two flagella, a short and long one. Though they are photosynthetic in the presence of sunlight, when deprived of sunlight, they behave like heterotroph by predating on other smaller organisms. Interestingly, the pigments of the euglenoid are identical to those present in the higher plant example, Euglena. 2.2.4 Slim molds. Slim molds are saprophytic protists. The body moves along decaying twig and leaves, engulfing organic material. Under suitable conditions, they form on the aggregation called plasmodium, which may grow and spread over several feet. During unfavorable conditions, the plasmodium different, differentiate, differentiate and form fruiting bodies, bearing spore at their tip. The spore possess true wall. They are extremely resistant and survive for many years. Even under adverse conditions, the spores are disappear by air currents. 2.2.5 Protozoans. All protozoans are heterotrophic and live as a predator or parasite. They are believed to be primitive relative of animal. There are four major groups of protozoan. First, amoeboid protozoan. These organisms live in freshwater, seawater, or moist soil. They move and capture their prey by putting out pseudopodia false feet, as in amoeba. Marine forms have silica shell on their surface. Some of them, such as entamoeba, are parasites. Second point, flagellated protozoan. The members of this group are either free-living or parasite. They have flagella. The parasitic form causes disease such as a sleeping sickness, example, trypanosoma. Third point, ciliated protozoan. These are aquatic, actively moving organisms because of the presence of thousands of cilia. They have a cavity gullet that open to the outside of the cell surface. The coordinate movement of row of cilia because the water laden with food to is strayed into the gullet. Example, Paramecium figure 2.4d. Sporozoan. These include diverse organisms that have a infectious spore-like stage in their life cycle. The most notorious in Plasmodium malaria parasite, which causes malaria or disease, which has a stagnating effect on human population. 2.3 Kingdom Fungi. The fungi constitute a unique kingdom of heterotrophic organisms. They show a great diversity in in morphology and habitat. You must have been fungi on the most bread and rotten fruit. The common mushroom you eat and toad tools are also fungi. White spots seen on a mustard leaf are due to a parasitic fungus. Some unicellular fungi, example yeast are used to make bread and beer. Other fungi causes disease in plant and animal. Wheat rust causing toxemia is an important example. Some are the source of the antibiotic example. Penicillium fungi are cosmopolitan and occur in air, water, soil and on animal and plant. They prefer to grow in warm and humid places. Have you ever wondered why are keep food in the refrigerator? Yes, it is to prevent food from going bad due to the bacterial or fungal infection. With the exception of yeast, which are unicellular, fungi are filamentous. Their bodies consist of long, slender, thread-like structure called hyphae. The network of hyphae is known as mycelium. Some hyphae are continuous tube filled with the multinucleated cytoplasm. These are called coinocytic hyphae. Other have separate or cross wall in their hyphae. The cell wall of fungi are composed of chitin and polysaccharide. Most fungi are heterotrophic and absorb soluble organic matter from dead substance and hence are called saprophyte. Those that depend on living plant and animal are called parasites. They can also live as symbiotic in association with algae as lichen and with root of higher plant as mycorrhiza. Reproduction in fungi can take place by vegetative means, fragmentation, fission and budding. Asexual reproduction is by spore called conidia or sporo, sporonog- Sporangiospore or zoospore and sexual reproduction is by the oospore, ascospore and basidiospore. The various spores are produced in the distinct structure called fruiting bodies. The sexual cycle involves the flowering following three steps. First, fusion of protoplasm between two motile or non-motile gamete called plasmogamy. Second point, fusion of two nucleus called karyogamy. Third point, meiosis in zygote resulting in the haploid spore. When a when a fungus reproduces sexually, two haploid hyphae of compatible mating type come together and fuse. In some fungi, the fusion of two haploid cells immediately resume in diploid cell to N. However, in other fungi, Ascomycetes and Basidiomycetes, on a intervening dikaryotic stage N plus N example, two nuclear per cell occur. Such a condition is called dikaryon, and the phase is called dikaryophase in fungus. Later, the parental nuclei fuse and the cell become diploid. The fungi form fruiting bodies in which reduction division occur, leading to formation of haploid spore. The morphological of the mycelium mode of spore formation and of fruiting bodies from the basic for the division of the kingdom into various classes. 2.3.1 Phycomycetes. Members of Phycomycetes are found in aquatic habitat and on decaying wood in most moist and damp places or as obligate parasitic on plant. The mycelium is aspected and zoonocytic asexual reproduction takes place by zoospore motile or by a planospore non-motile. These spores are endogenously produced in sporangium A zygospore is formed by fusion of two gametes. These gametes are similar in morphological isogamous or dissimilar in anisogamous or oogamous. Some common examples are Mucor, figure 2.5a, Rhizospores, the bread mold mentioned earlier, and Albugo, the parasitic fungi on mustard. 2.3.2 Ascomycetes. Commonly known as sec fungi, the Ascomycetes are mostly multicellular example, Penicillium, or rarely unicellular example, Yeast, Saccharomyces. They are saprophytic, decomposer, parasitic, or coprophilous growing on dung. Mycelium is branched and septate. The, the asexual spores are conidia produce exogenous on the special mycelium called conidiospore. Conidia on the germination produce mycelium sexual spore are called ascospore, which are produced endogenously in sac-like singular ascus. These ascus are arranged in different types of rotating bodies called the ascocarp. Some examples are Aspergillus figure 2.5b, Clavicapus and Neurospora. Neurospora is used extensively in biochemical and genetic work. Many members like morels and truffles are edible and are considered delicious. 2.3.3 Basidiomycetes. Commonly known form of basidiomycetes are mushroom, bracket fungi or puffball. They grow in soil or logs and tree stump and in living plant bodies as parasitic example rust and smut. The mycelium is branched and septate. The asexual spores are generally not found, but vegetative reproduction by fragmentation is common. The sex organs are absent, but plasmogamy is brought about the fusion of two vegetative or somatic cells of different strain or genotype. The resultant structure is dikaryotic, which ultimately gives rise to basidium. Karyogamy and meiosis take place in the basidium, producing four basidiospores. The basidiospores are exogenously produced on the basidium. The basidia are arranged in fruiting bodies called basidiocarp. Some common members are Agaricus, mushroom figure 2.5c, Ustilago, smut, and Puccinia, rust fungus. 2.3.4 Deuteromycetes, commonly known as imperfect fungi because only the asexual or vegetative phases of these fungi are known. When the sexual form of these fungi were discovered, they were moved into classes they are highly belong to. It is also possible that the asexual and vegetative stage have been given a one name and placed under Deuteromycetes, and the sexual stage another and placed under the another classes. Later, when the linkages were established, the fungi were correctly identified and moved out of the Deuteromycetes. Once perfect asexual stage of member of Deuteromycetes were discovered, they were often moved to Ascomycetes and Basidiomycetes. The Deuteromycetes reproduce only by asexual spore known as conidia. The mycelium is septate and branched. Some members are saprophyte or parasite, while a large number of them are decomposer or litter and help in mineral cycling. Some examples are Alternaria, Coletricum, and Trichoderma. 2.4 Kingdom Plantae. Kingdom Plantae include all eukaryotic chlorophyll-containing organisms commonly called plants. A few members are partially heterotrophic, such as the insectivorous plant and parasite. Bladderwort and Venus flytrap are examples of insectivorous plant, and Cusputa is a parasite. The plant cell have an eukaryotic structure with prominent chloroplast and cell wall mainly made of cellulose. You will study the eukaryotic cell structure in detail in chapter number 8. Plantae includes algae, bryophyte, pteridophyte, gymnosperm, and angiosperm. Life cycle of plant has two distinct phases: the diploid saprophytic and the haploid gametophytic that alternate with each other. The length of the haploid and diploid phases and whether these phases are free living or dependent on other vary among different groups in plant. This phenomena is called alteration of generation. You will study the further detail in the kingdom chapter number 3, Kingdom Animalia. This kingdom is characterized by heterotrophic eukaryotic organisms that are multinuclear and their cell lack cell wall. They directly or indirectly depend on plant for food. They digest their food in an internal cavity and store food reserve as a glycogen or fat. Their mode of nutrition is holozoic by ingestion of food. They follow a definite growth pattern and grow into adults that have a definite shape and size. Higher forms show a elaborate sensory and neuromotor mechanism. Most of them are capable of locomotion. The sexual reproduction is by copulation of male and female followed by embryological development. Salient feature of various phyla and described in chapter number 4, 2.6. Virus, viroids, prions, and lichens. In the five kingdom classification of Whitaker, there is no mention of lichen and some cellular organism like a virus, viroid, and prion. These are briefly introduced here. All of us who have suffered the ill effect of common cold or flu know what effect a virus can have on us, even if we do not asso- associate it with our condition. Virus did not find a place in classification since they are not considered truly living. If we understand living on those organisms that have a cell structure, the viruses are non-cellular organisms that are characterized by having a inert crystalline structure outside the living cell. Look at the picture. Once they infect a cell, they take over the machinery of the host cell, the replicate themselves, killing the host. Would you call virus living or non living? Virus means venom or poisonous fluid. Dmitry Levinsky, 1892, recognized certain microbes as causal organisms of the mosaic disease of the tobacco. Figure 2.6a. These were found to be smaller than the bacteria because they pass through the bacteria-proof filter. M. W. Bezernik, 1898, demonstrated that the extract of the infected plant of tobacco could causes infection in healthy plant and named the new pathogen virus and called the fluid of the contag- contagium vivum fluidium infection living fluid. WM silently 1935 showed that a virus could be crystallized and crystal consists largely a protein. They are inert outside their specific host cell. Viruses are obligate parasite. In addition to protein, viruses also contain genetic material that could be either RNA or DNA. No virus contain both RNA and DNA. A virus is a nucleoprotein and the genetic material in infectious. In generally, viruses that infect plant have single-stranded RNA and viruses that infect animal have either single or double-stranded RNA or double-stranded DNA. Bacterial viruses or bacteriophages viruses that infect the bacteria are usually double-stranded DNA virus figure 2.6b. The protein called a capsid made of a small subunit called capsomer. Protein protect the nucleic acid. In this capsomer are arranged in the helical or polyhedral geometrical form. Viruses cause diseases like mumps, smallpox, herpes, and influenza. AIDS in human is also caused by a virus in plant. The symptoms can be mosaic formation, leaf rolling and curling yellow, and vein clearing, rotting, and stunted growth. Viroid. In In 1971, T.O. Denier discovered a new infectious agent that was smaller than the virus and caused potato spindle tuber disease. It was found to be a free RNA. It lacked the protein code that is found in the virus, hence the name viroid. The RNA of the viroid was of low molecular weight. Prions. In modern medicine, certain infectious neurological diseases were found to be transmitted by an agent consisting of abnormally folded protein. The agent was smaller in size to viruses. These agents were called a prion. The most notable diseases caused by the prion are bovine spongiform encephalopathy (BSE), commonly called mad cow disease in the cattle, and its analogous variant, Cutis felid Jakob disease (CJD) in human lichen. Lichen are symbiotic association example mutually useful association between algae and fungi. The the algal component is known as the phycobionant and fungal component as mycobionant, which are autotrophic and heterotrophic respectively. Algae prepare food for fungi and fungi provide shelter and absorb mineral nutrition and water for its partner. So close is their association that if one saw in lichen in nature, one could never imagine that they have two different organisms within them. Lichen are very good pollution indicator. They do not grow in polluted area. Thanks for listening.